Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we are going to summarize all of the videos that we covered previously in chapters 1 to 7. If you are a healthcare student or professional, you must have a deep understanding of the human body. This means that to really understand the structure of a human body, what we call the anatomy, or the functioning of the human body, what we call physiology, this requires you to understand the smallest parts of the human body, and that is the biomolecules. So biochemistry and molecular biology, what we call BCMB, has now become a very important foundation for all of the other basic medical sciences, including cell biology, histology, anatomy, physiology, pharmacology, as well as pathology. In chapter 1, covered by videos 1 to 1.4, we emphasize the adult human body, which consists of about 30 trillion cells of 200 different types, 200 different specialized types of cells that we find in an adult human being. We then talked about what do these cells do, and one of the most important points we emphasize is energy. This is very important because, as you know, without energy, nothing can happen. The cell will be dead if it cannot or does not have enough energy. We also touch upon how this energy is made in cells, and that is by using a fuel and burning the fuel in the presence or absence of oxygen. So energy is a very important thing to keep in mind. It's required for all the 30 trillion cells, which means it's required for human survival and eventually for propagation of the species. And then in chapters 2 to 4, covered by videos 2.01 to 4.2, we discuss biomolecules. We talked about some of the major biomolecules that you find in the human body, and that included water, the inorganic chemicals such as the electrolytes, calcium, phosphate, iron, magnesium, bicarbonate, and so on. They were covered in 2.01 to 2.60 videos. Then we talked about an overview of organic chemicals in 2.70. And then we discuss carbohydrates, definitions that are used in carbohydrates, and there are many. Glycogen, glucose, A1C, which is a very important test nowadays for diabetes, and we'll talk more about this in future videos. We talked about identification, how carbohydrates are used for identification. We talked about ABO, blood groups, molecular recognition, glycocalyx, as well as proteoglycans in videos 2.71 to 2.79. And then we talked about lipids, including fatty acids, amphiphilic lipids, plasmalogens, steroids, eicosanoids, as well as wax esters. And these were covered in videos 2.8 to 2.86. Nucleotides and nucleic acids, including DNA and RNA, were covered in 2.90 to 2.92. And then we discussed amino acids and proteins in videos 3.0 to 3.13. And as examples of proteins, we talked about enzymes as well as enzyme kinetics in videos 4.0 to 4.2. One of the most important points that we emphasize in biomolecules is structure-activity relationship. How is the structure of a biomolecule ideal for its function? We use the analogy of the spoon, knife, and fork. A spoon has a certain structure because it has a certain function. A knife has a certain structure because it has a certain function. Similarly, a fork has a certain structure because it has a certain function. In the same way, all of these biomolecules have certain structures that are ideal to carry out their specific functions. So under biomolecules, we answered several important questions, such as what chemical properties of water makes it the solvent of choice? Why is concentration a very important thing to understand in understanding the human body? What are the rules of electrolytes and minerals in the body? Why is glucose the best fuel? Notice fuel is important for energy generation, followed by fatty acids and then proteins. What is the chemistry of glycogen that makes it an ideal glucose storage fuel? What's the chemistry of triglycerides that make it an ideal fatty acid storage fuel? Why are carbohydrates such good identification tags for cells as well as biomolecules? What is the chemistry of amphiphilic lipids that allows them to form membranes? What is in the structure of nucleic acids that make them ideal for information storage and transfer? And what is the chemistry of proteins that allows them to do so many things for cells, such as increasing reaction rates that we see with enzymes, binding oxygen that we see in myoglobin and hemoglobin, regulating information transfer that we see with transcription factors, protecting the body that we see with proteins such as mucin, antibodies, and several other defense proteins. So these topics were covered in videos or chapters 2 to 4, videos 2.01 to 4.2. And then we went on to chapter 5. And the most important question in chapter 5 is this: how does our body generate energy? Remember, without energy, nothing is going to happen. The cell will die. So how does our body generate energy at all times, 24/7, since we do not store energy? Since we use energy in the form of ATP, we do not store ATP. We mentioned that if you are 150 pounds, you probably need about 150 pounds of ATP per day. But at any one time, if we measure the amount of ATP in your body, you probably have less than 0.1 pounds. So we don't store energy, we make energy as we need it. So this is a very, very, very important point for the healthcare professional to appreciate. We don't store energy, we have to continuously make energy. The more we need it, the more we have to make it. Since we are made up of 30 trillion cells, this question basically becomes, how do these 30 trillion cells generate energy at all times, since they do not store energy? Another important point we emphasize when we talk about these 30 trillion cells is that 24 trillion cells, okay, so that's about 80%, that's 24 trillion cells, are actually red blood cells, and then the remaining 6 trillion cells are the 199 other types. Okay, so this is an important statistic to keep in mind as we go through biochemistry and your rest of your biomedical sciences. As we mentioned, if the cell doesn't have energy or insufficient energy, that is going to cause cell damage and eventually cell death. And therefore, it's not surprising that many important diseases in medicine affect energy generation. And this includes cardiovascular disease such as myocardial infarction and stroke, cancer, diabetes, and all kinds of pulmonary diseases. So this question was answered in Chapter 5, videos 5.0 to 5.30, where we talked about metabolism, which is basically a combination of anabolism, which is synthesis, and catabolism, which is breakdown. In this chapter, we discuss ATP generation. How is ATP made? We discuss how cells use the most important fuel, glucose, to make ATP by what we call the central pathway that included glycolysis, the pyruvate dehydrogenase complex, the Krebs cycle, electron transport chain, and oxidative phosphorylation. We also discussed inhibitors and uncouplers, the chemicals that can inhibit or uncouple the electron transport chain from oxidative phosphorylation. Rather than simply going through metabolism, we discussed more of a practical application of metabolism. We first talked about postprandial state, and this is after a meal. And under this state, we talked about glycogenesis, synthesis of glycogen, fatty acid synthesis, triglyceride synthesis, amphiphilic lipid synthesis, cholesterol synthesis, the hexose monophosphate shunt, as well as amino acid synthesis. Under this state, we also talked a little bit about the absorption of lipids as well as proteins. Then, under starvation, we talked about the metabolic pathways that predominate under starvation, and that includes glycogen breakdown or glycogenolysis, breakdown of fatty acids or beta oxidation, fatty acid oxidation, breakdown of proteins, autophagy, as well as the one synthesis that happens during starvation, and that's the synthesis of glucose, gluconeogenesis. So as we mentioned, this is a very important question, continuous energy generation for the healthcare professional. And energy is generated by burning fuel in the presence or absence of oxygen. We saw in Chapter 5 that if we had oxygen, you'll get more energy when you burn the fuel, but you don't have to have oxygen. You can still get energy directly from fuel, but in that case, you get less energy. We did not talk much about oxygen supply because that's more of a physiological concept, but it's important for you to know that oxygen comes through your lungs and then goes to the heart, eventually picked up by the red blood cells in blood vessels. So these structures here are important for oxygen absorption into the body and delivery to all parts of the body. So it's not surprising that anytime there is impaired oxygen supply to the tissues, that's going to affect energy generation. And this is what we see in various diseases that affect either the lungs, for example, pulmonary disease, heart disease, if there is not enough red blood cells in anemia, or if the blood vessels are blocked, like in arteriosclerosis. All that is going to affect oxygen supply. In Chapter 5, we assume there was adequate oxygen supply. So under these circumstances, then the question now becomes, how does the body supply fuel to all 30 trillion cells all the time. If there is enough oxygen, the question now is, how does the body send fuel to all the 30 trillion cells all the time? Now, the major fuels are glucose and fatty acids. So it's important to note, as we discussed in chapter 5, that most cell types can use glucose or fatty acids for energy, for ATP generation. However, and this is important to note, a few cell types can only use glucose for energy. So this includes the 24 trillion red blood cells that can only use glucose, they cannot use fatty acids, and the brain cells, they use predominantly glucose. They get their ATP from glucose predominantly, 80% glucose. A little bit of ATP can come from fatty acids, but no more than 20%. So the brain relies very heavily on glucose supply from the bloodstream. This means that the concentration of glucose in the bloodstream must always be sufficient to supply the red blood cells, but more importantly, the brain, in order to meet their energy generation needs. So for the healthcare professional, it's important to remember our blood glucose concentration must always be greater than 40 to 50 milligrams per deciliter, because only under this concentration will the brain be able to extract enough glucose from the blood to provide it with energy. If you have less than this, we will start seeing very serious symptoms, what we call hypoglycemia, very low glucose concentration. That's going to affect the brain. We're going to have symptoms like trembling, weakness, dizziness, seizure, loss of consciousness, and even coma as well as death. So this is a very important point for healthcare professionals to remember. We must keep our blood glucose concentration more than 40 milligrams per deciliter to feed our brain cells. Okay, this is a critical blood glucose concentration. Most hospitals use around 40 to 50 milligrams per deciliter as a critical value for our blood glucose. So as we went through metabolism after a meal, postprandial, as well as starvation, whether it's early starvation or late starvation, this was one very, very important point that we keep in our mind, and that is, how is the body maintaining its blood glucose to make sure it's greater than 40 milligrams per deciliter. So when you are eating a meal, a normal diet, by normal we mean a diet that has carbohydrates. We're not talking about specialized diets like the Atkins diet that doesn't have carbohydrates. Okay, so a normal diet. So if we eat a normal diet that has carbohydrates, that should not be a problem, because the glucose from our diet will be absorbed into our bloodstream, and that will keep the blood glucose concentration greater than 40 milligrams per deciliter. So after a meal, a normal diet, the glucose from the diet from the GI tract will enter the body, and it will keep our blood glucose concentration more than 40. This typically goes more than even 100 milligrams per deciliter, which means there's more than enough glucose for every cell to use glucose to generate ATP. So the liver will use glucose to generate ATP, the red blood cells, the brain cells, the adipocytes, as well as the muscle cells and all the other cells. So everyone basically will use glucose to generate energy. After a meal, the major regulation hormone is insulin, And we see a lot of synthesis happening because there's enough energy from glucose. So the adipocytes will synthesize lipids, the muscles will synthesize glycogen as well as proteins, the liver will synthesize glycogen as well as proteins as well as lipids. So a lot of synthesis happens, and that's one thing insulin does. It stimulates a lot of synthesis pathways. So after a meal, maintaining blood glucose is not a problem. However, how do we maintain our blood glucose when we are not eating? For example, we are fast asleep, 2 o'clock, 3 o'clock in the morning, or we are lost in the woods, or we simply do not have access to food. So how is our blood glucose concentration maintained during starvation times? And we saw in chapter 5 when there is no glucose coming from the diet. So when starvation begins, the liver plays a very important role. It breaks down its glycogen and it also makes a little bit of glucose. And this glucose from the liver is what will maintain our blood glucose concentration. So the liver will be maintaining our blood glucose concentration. During this time, insulin levels are low, but several other hormones start increasing, especially glucagon, catecholamines, cortisol, as well as growth hormone. So these hormones stimulate catabolism, so we're going to see a lot of breakdown. So for example, there is fatty acid breakdown in the liver, because the liver will now start using fatty acids for ATP. There'll be fatty acid breakdown in the adipocytes. There'll be triglyceride breakdown in the adipocytes, and the muscles also will use fatty acids for their ATP, so that this glucose that is coming from the liver will be reserved more for the red blood cells as well as the brain. So at this point in time, the brain as well as the red blood cells will use glucose for ATP generation, while the other tissues will use fatty acids for ATP. Now, if starvation continues, then early starvation will become late starvation. And here the cutoff really is glycogen. Once the liver glycogen starts depleting, we slowly enter into late starvation. And we in chapter 5 we divided late starvation, just for understanding late starvation, into different phases. We started off with phase 1. So now that there is no more glycogen in the liver, the liver cannot break down glycogen to maintain blood glucose. So now it resorts more to synthesis of new glucose, gluconeogenesis. So the liver makes quite a bit of glucose by gluconeogenesis, but there are several other organs that help. The kidneys will also make glucose by gluconeogenesis, as well as the GI tract. So this glucose from the liver, kidneys, and GI tract will help maintain blood glucose concentration, and that glucose will now help the red blood cells as well as the brain for ATP generation. As the liver starts using gluconeogenesis to make glucose, we also see protein breakdown, protein catabolism in the muscles, because this provides the raw materials for the liver to make glucose. So protein catabolism becomes predominant during this time. So catabolism in general, so once again we see fatty acid oxidation, breakdown of fatty acids, breakdown of lipids in adipocytes as well as in muscles, and there's also glycogen breakdown in muscles, as well as fatty acid breakdown in liver to generate ATP. So once again, these hormones, glucagon, catecholamines, cortisol, and growth hormone, they basically stimulate catabolism. So under these conditions, now the brain red blood cells, as before, use glucose for ATP, while other tissues use fatty acids. So this is late starvation, and if late starvation keeps on continuing, then we are going to have a problem, and that is we are breaking down more and more of our muscle proteins, because this provide the raw materials necessary for the liver to make glucose. Not just the liver, but also the kidneys, as well as the GI tract. So we don't want our muscles to keep on breaking down under very late starvation conditions, because then we'll be too tired to go find food. So we want this to stop. So then we enter what we termed phase 2. And in late starvation phase 2, now the liver not only makes glucose by gluconeogenesis, but it does something very important. Now this pathway, synthesis of ketone bodies, become more and more predominant. So ketone bodies are made from fatty acids, so the liver starts making ketone bodies. Now why does it do that? Because ketone bodies are water soluble, and most importantly, they can be used by the brain to generate ATP. So under these conditions, the brain can now use glucose as well as ketone bodies to generate ATP. Now the red blood cells, however, cannot use ketone bodies, so they still rely on glucose for energy generation. However, since the brain can use ketone bodies, so they don't need as much glucose as before, as in phase 1. So now we see this protein catabolism slow down. So it buys us more time to find food in order to survive. So phase 2 is quite similar to phase 1, except for the fact that now ketogenesis, synthesis of ketone bodies, predominates. Becomes more important, and ketone bodies and glucose can be used by the brain, while other tissues can use fatty acids or ketone bodies for ATP generation. So to summarize this very important question for the healthcare student and professional to remember, what maintains your blood glucose concentration? So after a meal, it's our glucose from the diet. The major hormone that is plays a role here is insulin. Leptin is the hormone that will, after you eat, it will tell you that you are full. So leptin is a hormone that also plays an important role. During starvation, in early starvation, liver glycogen breakdown as well as liver gluconeogenesis. In late starvation, when there's no more liver glycogen, then liver gluconeogenesis, small intestine gluconeogenesis, and kidney gluconeogenesis. So these are the pathways and the organs involved in maintaining our blood glucose concentration. And during starvation, these are the hormones that play an important role, including ghrelin that will be released when we are hungry. So this is a good summary to keep in mind, how we must make sure that we always keep our blood glucose concentration above this critical value of 40 to 50 milligrams per deciliter. After answering this question in chapter 5, then in chapter 6, we talked about how is metabolism regulated so that Anabolism predominates after having a meal, while catabolism will predominate during starvation in order to make sure that all cells in the body have sufficient ATP. So not only is it important for us to carry out metabolism to make sure that all our cells have enough energy, but it's also very important to regulate metabolism. So under chapter 6, which is video 6.0 to 6.19, we talked about regulation of protein function as well as location. We talked about how we can do this by sequestering enzymes. We talked about cofactors, allosteric regulation, as well as covalent modifications. We talked about zymogens as well as isoenzymes. And then we discussed how we can regulate the actual amount of proteins in order to regulate metabolism. We then talked about metabolism regulation after a meal, especially the role of insulin. And then we talked about metabolism regulation during starvation, and there the role of glucagon, catecholamines, cortisol, as well as growth hormone. And we also discussed these two important hormones, ghrelin and leptin. Since these hormones are basically signals for cell signaling, the next chapter, which is chapter 7, we talked about cell signaling, and there the important question is, besides metabolism, how is everything else, such as development, immune response, cell growth, differentiation, nervous system function, and so on, how is everything else regulated? And this is what we discussed in chapter 7, video 7.0 to 7.4, how do cells talk to one another. So we talked about types of cell signaling, we talked about various signals, including hormones, and then we talked about the different types of receptors, including the ligand-gated ion channel receptors, G-protein coupled receptors, enzyme-linked receptors, as well as intracellular receptors. So this is a summary of the 7 chapters that we have covered so far that will help us understand future videos when we talk about topics such as cell division and cancer, as well as specific biochemistry of tissues, as well as looking at certain topics in pathology and certain biochemical aspects of drugs in pharmacology. Until the next video, thank you for supporting this channel and have a wonderful day. We'll see you next time.